I want you to imagine, just for a second, that you're a doctor trying to hunt down an enemy.
Okay, setting the scene.
Right. But, I mean, this isn't just any enemy. This one is so heavily armored, like perfectly shielded, that it's completely invisible to all of your standard diagnostic defenses.
Oh, wow.
Yeah, you know it's there in the patient, you know it's lethal. But when you look through your microscope, there is absolutely nothing to see. So how do you track down a killer that just refuses to show its face?
It really sounds like the premise of a medical thriller. But for clinicians dealing with some of the most dangerous pathogens on Earth, that invisibility is just a daily biological reality.
Right, because if you can't see the pathogen, you can't definitively diagnose the illness.
Exactly. Which means you are essentially flying blind when it comes to treatment.
You shared this massive stack of microbiology sources with us about this very problem. And today's mission is to figure out how doctors actually unmask these invisible killers.
Yes, it's a fascinating topic.
We're doing a deep dive into a concept called acid-fast staining. We're going to explore how a laboratory trick engineered all the way back in the 1880s uses heat, a splash of harsh acid, and some very clever chemistry to just break right through the toughest bacterial armor on the planet.
And the sources you provided really offer a fantastic lens into all this. We'll be looking at the underlying biochemistry of the staining technique, the sheer desperation in the 19th century that led to its invention, and some incredibly surprising modern applications.
Applications that go way beyond just bacteria, right?
Right, way beyond. And perhaps most importantly, we're going to examine why this decidedly low-tech, century-old method is still holding its ground today, even in the era of advanced DNA testing.
Okay, let's unpack this, because before we get into the chemical hacks used to die these bacteria, we really need to establish why they're so incredibly stubborn in the first place.
It's a good place to start.
So normally, if a microbiology lab wants to visualize a bacterium, they use a standard gram stain.
Right, the go-to method.
But doing a gram stain on these particular bugs, specifically the genus Mycobacterium, is a lot like trying to paint a car that has just been heavily waxed.
Yeah, that's a perfect analogy. Because just like a freshly waxed car, when lab technicians throw a standard water-based dye at a mycobacterium, like the crystal violet used in a gram stain, the dye just beads up.
It just rolls right off.
Exactly. The water-based pigment literally cannot physically grip or penetrate the surface.
So when you look through the microscope, instead of seeing clearly defined, brightly colored cells, you just see these faint, colorless voids amidst all the other stained cellular debris.
And the clinical term in the sources for these voids is actually ghosts.
Wait, ghosts?
Yeah, you are literally looking at nearly invisible ghost cells. And to understand why that happens, you really have to look at the extreme architecture of their cell wall.
Because most normal bacteria have a wall made primarily of peptidoglycan, which is like a sugar amino acid polymer.
Right. Think of it like a chain link fence.
But mycobacteria are built entirely differently. They're essentially wearing a biochemical suit of armor.
And the specific chemical makeup of that armor is what makes them so formidable. The foundation is still a thin layer of peptidoglycan, but that is anchored to a complex carbohydrate called arabinogalactan.
Okay.
So if the peptidoglycan is the concrete foundation, the arabinogalactan is like the steel rebar sticking out of it.
That makes sense.
And attached to that rebar is a remarkably thick, dense layer of what are called mycolic acids.
And these mycolic acids are the wax, right?
Mm-hmm.
I read in the sources they are massive fatty acid chains, something like 60 to 90 carbons long.
They're enormous, just highly branched lipid molecules, and this creates an incredibly dense, hydrophobic, meaning water-repelling scaffold around the entire cell.
And this waxy fortress isn't just, an annoyance for laboratory technicians trying to stain it. It's actually the core reason these bacteria are so deadly.
Precisely. That wall makes them notoriously resistant to drying out, meaning they can survive on surfaces or in airborne droplets for really long periods.
And it makes them resistant to standard chemical disinfectants that would normally just burst a normal bacterium.
Right. And most critically, it allows them to survive our own immune system.
Oh, yeah. I read the section on macrophages, and it is genuinely unsettling.
Yeah, really.
Because a macrophage is one of our body's primary immune cells. It's sort of a cellular garbage disposal that swallows up invading bacteria and dissolves them with enzymes.
Right, that's its whole job.
But when a macrophage swallows a mycobacterium, it completely fails.
Yeah, the macrophage engulfs the bacterium, pulling it into a highly acidic internal compartment, and then it pumps in digestive enzymes.
But the enzymes just bounce off that waxy mycolic acid shield.
Exactly. The mycobacterium doesn't just survive, it actually sets up camp, happily living and multiplying inside the very immune cell that was supposed to destroy it.
Now, speaking of acidic environments, I have to jump in with a point of confusion that hit me when I first opened these sources.
Oh, what was that?
So the entire technique is called acid-fast staining. And my immediate assumption was that acid-fast meant these organisms love acid. Like they thrive in highly acidic environments, maybe hot springs or human stomach acid.
You know, that is a very common intuitive leap, but the term acid fast has absolutely nothing to do with the organism's lifestyle or its preferred environmental pH.
So it has nothing to do with surviving inside that acidic macrophage either.
Not directly, no. It is purely a description of how that waxy cell wall behaves chemically in a laboratory test tube. The word fast here is used in the Old English sense of the word, meaning to hold firmly or to be steadfast.
Oh, like a ship being held fast by an anchor.
Exactly, or a dye in clothing being color fast so it doesn't wash out in the laundry.
Ah, okay. So the bacteria are holding fast to the dye. They refuse to let go of the color, even when you try to forcefully wash it away with acid.
Precisely, and while we are clearing up misconceptions in the terminology, the sources addressed another major one.
Right, about them being gram-negative.
Yeah, because these mycobacteria repel the traditional gram stain, people historically assumed they must be gram-negative bugs. But based on the actual biochemical structure we just talked about, that inner membrane, and the peptidoglycan-linked layer, microbiologists technically classify them as atypical gram-positive.
Atypical gram-positive.
Oh.
Okay, so we're dealing with atypical armored ghost bugs that laugh at water-based dyes and use our own immune cells as hotels.
That's one way to summarize it, yeah.
If standard dyes just bounce right off this wax, I mean, somebody had to figure out a chemical workaround to finally breach it.
And to appreciate how they cracked that code, you really have to look at the sheer desperation of the 1880s. The medical community was facing an unimaginable crisis.
The sources paint a pretty grim picture. Tuberculosis was wiping out roughly 1 in 7 people across Europe and the United States.
1 in 7. Just let that sink in.
It's terrifying.
It was a global public health catastrophe. Doctors desperately needed a way to confirm an infection quickly from a patient's sputum sample.
Because at the time, if a patient came in coughing, you essentially had to wait for the disease to progress to diagnose it, right?
Yeah, and by then, the patient was usually doomed and had already infected their entire family. But then, on March 24th, 1882, Robert Koch stood up in front of the Physiological Society of Berlin and made a monumental announcement.
He had successfully identified the specific microscopic culprit Mycobacterium tuberculosis.
Exactly.
And that date is why we still commemorate March 24th as World TB Day.
Right.
But as massive as Koch's discovery was, his actual staining method was practically useless for an everyday clinic.
Right, it was just too slow.
It involved soaking the sample in an alkaline dye for 20 to 24 hours. No busy doctor has time for that.
Koch found the bug, but he didn't invent the rapid diagnostic tool. The person who figured out the core mechanism of acid fastness is often totally overshadowed in this specific story.
Paul Ehrlich.
Yes, Ehrlich was actually sitting in the audience during Koch's presentation. He was already obsessed with chemical dyes, and within months, he noticed something incredible.
What did he do?
He developed a method using an aniline dye combined with heat, and he found that once he forced the dye into the tubercle bacillus, it violently resisted being washed out, even when he poured dilute mineral acids right over it.
So Ehrlich literally documented the very first observation of the acid-fast property. He proved the wax could be breached.
He did. But Ehrlich's method, while a scientific breakthrough, was still just too clumsy and inconsistent for routine hospital use.
Which is where Franz Seele and Friedrich Nielsen enter the picture. Working around 1882 and 1883, they took Ehrlich's raw concept and engineered it into a rugged, reliable tool.
Right. Seele had the brilliant insight to add carbolic acid, which we now call phenol, to the dye mixture.
And phenol is a lipid-soluble organic compound, right?
Exactly. It essentially acts as a chemical Trojan horse, allowing the red dye to dissolve directly into the waxy lipids of the bacterial wall.
That is so smart.
And then Nielsen standardized the exact heating temperatures and the concentration of the acid alcohol wash.
And just like that, the Seele-Nielsen stain is born. It's really fascinating that the two men whose names are forever attached to the technique didn't discover the bacterium or the acid-fast property itself.
They were just the master chefs who perfected the recipe.
But that recipe is a masterpiece of biochemical logic. It uses a combination of physical brute force and chemical solvent properties to defeat the armor. And here's where it gets really interesting.
Oh, the actual staining process.
Yeah, let's walk through how this actually works on a slide, because it honestly feels like a heist movie.
It really does.
So you have a glass slide smeared with a patient's sample. First, you flood it with this phenol-based red dye called carbofusin, but the wax is still repelling it. So how do they force the door open? The sources mention steaming to flame.
Right. You literally light a Bunsen burner and heat the underside of the glass slide until the dye starts to steam.
You're just cooking it.
Essentially, yeah. Heat increases the kinetic energy of those massive mycolic acid chains in the cell wall, so the waxy scaffold physically loosens and expands, creating microscopic gaps.
And that's when the Trojan horse strikes.
Exactly. The phenol pulls the bright red carbofusin dye straight through those gaps and deep into the cell.
Okay, so you've melted the wax and forced the dye inside. You remove the heat, the slide cools down, and the wax solidifies again, trapping all that red dye inside the cell like a vault locking shut.
A perfect heist.
But here's the problem. You just heated the whole slide. Every harmless skin cell, every piece of mucus, every normal mouth bacterium on that slide is also stuffed full of red dye.
Right.
If you look at it now, everything is just a giant red blur. So how do you isolate the killer?
This is where the brilliant second step comes in, the decolorizing wash. You flood the slide with a harsh solution of roughly 3% hydrochloric acid mixed in ethanol.
That sounds intense.
It is. For almost every other normal cell on that slide, this acid alcohol is devastating. The alcohol dehydrates their normal cell walls, and the acid easily drops the pH, breaking the ionic bonds holding the red dye.
So all those normal cells are instantly stripped of their color. They go completely invisible again.
But the mycobacteria don't. Because they are wrapped in that dense hydrophobic wax, the acid-alcohol mixture literally cannot penetrate the armor to wash the dye out.
The dye is locked in the vault. They hold fast against the acid.
Exactly. And because everything else on the slide is now invisible, you need a way to see the background so you know you are actually in focus. So you apply a counterstain, usually a simple blue dye called methylene blue.
So the normal non-acid fast cells, which were just stripped naked by the acid, easily soak up the blue dye.
Yes, and the acid-fast mycobacteria, completely full of red dye and protected by their wax, they just ignore the blue.
The resulting visual is stunning. If you look at the images in the sources, you see this broad sea of blue background material, blue skin cells, blue immune cells, and right in the middle of it, these striking, unmistakable, slender, bright red rods.
It is arguably one of the most iconic images in all of microscopic medicine. You melt the armor, sneak the red dye in, lock the armor back up, and wash everything else away with acid.
That red-on-blue image is famous worldwide for diagnosing tuberculosis. But reading further into the sources, I mean, the big revelation for me was that this chemical trick isn't exclusive to TB.
Not at all.
Acid fastness is a property of the wall, not a VIP club for one specific disease.
And that is a crucial distinction in microbiology. Any organism that happens to synthesize a similarly waxy, lipid-rich cell wall will hold on to that red dye.
Let's look at another terrifying disease, leprosy, clinically known as Hansen's disease.
Right. Caused by Mycobacterium leprae.
The sources highlight a mind-blowing fact about this bug. It grows so incredibly slowly and has such a strict preference for cooler body temperatures that in 150 years since it was discovered, scientists have never been able to grow it on a standard petri dish in a lab.
It is entirely unculturable on standard artificial media.
That's wild.
Yeah, to study Mycobacterium leprae, researchers are actually forced to grow it inside the footpads of mice, or in 9-banded armadillos.
Armadillos? Wait, really?
Yes. Because those animals have core body temperatures slightly lower than a human's, which the bacteria strictly require to multiply.
But because doctors can't just swab a patient, put it on a warm gel, and wait for a colony to grow to see what they are dealing with, they still rely heavily on the acid-fast stain.
They absolutely do.
They take slit skin smears from a patient's earlobe or elbow, stain them, and then literally just count the red bacilli under the microscope.
Clinicians use a standardized metric called the Ridley-Jopling scale. It is a semi-quantitative bacterial index.
Okay, so it's all about density.
Right. They determine the severity of the leprosy, classify the specific type, and monitor whether the antibiotic therapy is actually working over months of treatment. And that's based almost entirely on the physical density of those red acid-fast rods they can spot per microscope field.
Okay, so TB and leprosy both use it. but they're both mycobacteria. I want to bring up the 1993 Milwaukee water crisis because this completely broke my mental model of what this stain is used for.
Oh, this is a great example.
The sources mention that 400,000 people in Wisconsin got violently ill from the municipal water supply.
Yeah.
But the culprit wasn't a bacterium at all. So why were public health officials running bacterial stains on a city water supply?
Well, the pathogen in Milwaukee was Cryptosporidium parvum. It is a protozoan parasite.
Which belongs to a completely different kingdom of life than bacteria.
Exactly.
Hmm.
However, during its life cycle, it forms these hardy, spore-like structures called oocysts, which it sheds into the environment. And the protective outer wall of those oocysts happens to be packed with complex lipids that behave remarkably like mycolic acid.
So it's convergent evolution. A totally different organism built a similar waxy shield.
Bingo.
And because of that, if you run a modified acid-fast stain using a slightly weaker acid so you don't completely obliterate the parasite, those Cryptosporidium oocysts just light up.
They do.
The sources describe them showing up as vivid pinkish-red spheres against a blue or green background of stool debris.
It highlights perfectly that acid fastness is a structural spectrum, not a binary bacterial label. You have completely non-bacterial parasites like Cryptosporidium that possess the trait.
Right.
You also have other bacterial genera like Nocardia that are considered partially acid fast.
Partially. How does that work?
Well, Nocardia has shorter mycolic acid chains, so it can survive a weak acid wash. But if you hit it with the aggressive hydrochloric acid used for TB, the dye washes right out.
Okay, that makes sense. But this brings me to my biggest point of contention with the sources and really with modern medicine in general.
Okay, let's hear it.
We've established this is a brilliant, rugged 19th century chemistry hack. But we live in the 21st century. We have robotics, we have artificial intelligence, we have DNA testing.
You do.
The sources talk about machines like the GenExpert MPBRIF assay. It takes a sample, amplifies the exact DNA of the tuberculosis bacteria, and tells you in 2 hours if the patient has TB and whether that specific strain is resistant to first-line antibiotics.
The GenExpert is an absolute marvel of molecular diagnostics. The World Health Organization endorsed it back in 2010 for a very good reason.
That's super precise, right?
It is vastly more sensitive than a human looking through a microscope, meaning it can detect an infection even if there are only a few dozen bacteria present in the whole sample.
Right. So what does this all mean? If we have a machine that gives us the exact DNA footprint and the antibiotic resistance profile in 2 hours, using a 140-year-old microscope trick just seems reckless. Why hasn't the global health community completely banned the Seal-Nielsen stain and replaced it with these machines?
Because global health is ultimately dictated by economics and infrastructure. In a perfect, fully funded world, sure, every clinic would have a GenExpert.
Yeah.
But here is the stark reality. A single expert testing cartridge costs roughly $10.
$10.
And that is with heavily subsidized, not-for-profit pricing negotiated for developing nations. Furthermore, the machine requires a climate-controlled room, rigorous annual calibration, and a completely stable electrical grid.
Yep.
If the power cuts out during that 2-hour run, the $10 cartridge is completely ruined.
Wow. So $10 per test plus thousands for the machine is just an impossible barrier in a rural clinic in sub-Saharan Africa or parts of Southeast Asia.
It really is. Contrast that with the classic Seal-Nielsen stain. What does that cost?
I think the sources said less than 50 cents per slide in materials.
Exactly. Less than 50 cents. It requires nothing more than a glass slide, some stable chemical dyes, a flame, and a basic light microscope powered by a mirror reflecting sunlight if necessary.
That's incredible. There is a middle ground mentioned in the sources, though. Fluorescence microscopy.
Yes, the auramine-rhodamine stain.
Right.
Instead of using a red dye, you use a fluorescent dye that binds to the wax. Under a special microscope, the mycobacteria glow a brilliant apple green against a dark background.
That sounds way easier to see.
It is highly recommended by the WHO because a technician can scan the slide at a much lower magnification. The bright green pops out instantly, making it much faster to read than searching for tiny red rods.
But I'm guessing you hit an infrastructure wall there, too.
Exactly. Fluorescence microscopes are expensive to buy, the specialized bulbs burn out and are costly to replace, and they require a completely dark room.
So we are stuck relying on the cheap 50-cent 19th century test for much of the world. But the sources are very clear that this old-school method has massive, dangerous pitfalls.
It definitely does.
A positive red-on-blue smear does not guarantee the patient actually has tuberculosis.
And this is the danger of a test that identifies a type of cell wall rather than a specific species' DNA. There is an entire category of bacteria called non-tuberculous mycobacteria, or NTMs.
Right.
A classic example is the Mycobacterium avium complex. They are literally everywhere in the environment, in the soil, in the water.
And these are opportunistic pathogens. They don't usually make healthy people sick, but they attack people whose immune systems are already severely compromised.
Exactly. And this caused a major diagnostic tragedy during the height of the HIV/AIDS epidemic in the 1980s and 1990s. Yeah. A severely immunocompromised patient would come into the hospital with severe respiratory failure. The clinic would run a rapid acid-fast smear, see the red rods, and immediately assume it was aggressive tuberculosis.
But the stain can't tell the difference.
Right, because the stain cannot distinguish between Mycobacterium tuberculosis and a harmless environmental NTM like Mycobacterium avium. Patients were frequently misdiagnosed.
So they were getting the wrong treatment.
They were put on toxic TB drug regimens that did absolutely nothing for their actual NTM infection.
Which means you absolutely need a culture or a DNA test to confirm the exact species. But if we're talking about pitfalls, we have to talk about what is easily the most counterintuitive, head-scratching failure mode in all the sources.
The dead bug paradox.
Yes, the dead bug paradox. It is a fascinating quirk of how antibiotics actually work versus how the stain works.
It really is.
Because the acid-fast stain cannot tell you if the organism you are looking at is actually alive.
When you give a patient modern antibiotics for tuberculosis, those drugs generally don't work by dissolving the external waxy wall.
Right, they go inside.
Exactly. They penetrate the cell and target the internal machinery. They stop the bacteria from synthesizing RNA, or they destroy the internal ribosomes. So the cell dies from the inside out.
But that dense, waxy, mycolic acid armor on the outside, it remains structurally intact for weeks, sometimes months, after the bacterium is totally dead.
So you could have a patient who has been on heavy TB medication for a month. The drugs are working perfectly. The infection is completely cleared.
But if you take a sputum sample and run an acid-fast stain,
you will still see hundreds of bright red rods. The dead, hollowed-out, waxy shells hold on to the red dye just as fiercely as living cells do.
that is wild.
Historically, this terrified doctors. They would see a strongly positive smear weeks into therapy and just panic, assuming the antibiotics were failing.
Which led to them changing treatments.
Yes. This led to doctors unnecessarily changing the patient's drug regimen, risking severe side effects, all because they were being tricked by microscopic fossils.
The fossil of the bacteria holds the dye. That perfectly explains why the sources emphasize that, despite all our technology, the old-school, agonizingly slow culture methods are still the absolute gold standard for confirming if a patient is actually cured.
Right. Methods like the Lowenstein-Jensen culture.
How does that work?
You take the patient sample, put it on this specific solid nutritional gel made with egg and potato, and you wait to see if the bacteria actually multiply and grow into physical colonies. If they grow, they're alive. If nothing grows, they're dead.
But because Mycobacteria expends so much metabolic energy building that massive waxy armor, they divide incredibly slowly, right?
Or slowly.
The sources said their generation time is 15 to 20 hours compared to maybe 20 minutes for something like E. coli.
Yeah.
And because of that, those culture results can take anywhere from 4 to 8 weeks to appear.
Which is an agonizingly long time to wait when you are dealing with a lethal, highly contagious airborne disease.
And that brings us full circle to why this 140-year-old chemical trick remains globally vital today. It bridges the gap between waiting 2 months for a culture and spending millions on DNA infrastructure.
Exactly.
It spans from tracking down waterborne protozoa in major modern cities like Milwaukee to diagnosing leprosy in the Amazon.
And if we bring it right back to you, the listener, the relevance is immediate.
How so?
Let's say you live in a wealthy city with a state-of-the-art hospital, and a patient comes into the emergency room coughing up blood. You don't have 2 hours to wait for the DNA test to run, and you certainly don't have 8 weeks to wait for a culture gel to grow.
Right, you need answers now.
An acid-fast smear takes minutes. It is the ultimate fast triage step. If the lab tech looks through the microscope and sees those striking red rods, it's an immediate, massive red flag.
It prompts the hospital to instantly put that patient into a negative pressure airborne isolation room.
Exactly, protecting the nurses, the doctors, and everyone else in the waiting room long before the fancy genetic results confirm the details.
It is the ultimate first line of defense, born from 19th century desperation. We started this deep dive by talking about an invisible, heavily armored enemy.
We did.
But the fact that its armor, that incredible water-repelling, acid-resisting waxy wall, remains perfectly intact and detectable as a microscopic fossil long after the organism itself is completely dead. I mean, it leaves me with a somewhat haunting question to ponder.
What's that?
If these microscopic waxy fortresses take so long to break down in the human body, what other invisible fossilized remnants of microscopic life might we be interacting with, or even breathing in, every single day without ever realizing they're there?